I would like to thank the organizer for giving me this opportunity to share our recent work on long cell-free DNA in this meeting. So our group has a long-standing interest in the development of novel molecular diagnostic based on cell-free DNA in the plasma, especially for its application in pregnancy and cancer. It is well known that cell-free DNA is released into the circulation upon cell death. So unlike the intact cellular DNA, cell-free DNA in the circulation is highly fragmented. So here shows a typical size profile of plasma DNA that we can obtain from a short-read sequencer. And from this fragment size profile, we can see a predominant peak at 166 base pair, a minor peak at 143 base pair, and a 10 base pair periodicity below 143 base pair. And it has been suggested that for fragments of 143 base pair, it actually resembles DNA wrapped around the histone core. And for the fragments of 166 base pair, it actually resembles DNA wrapped around the histone core plus a 20 base pair linker. And for the 10 base pair periodicity, it actually resembles the patterns of nuclease cleavage of the nucleosome-bonded DNA. So in the field of liquid biopsy, people have been focusing on the analysis of short cell-free DNA. But just a few years ago, a group started to wonder whether these short cell-free DNA represent only the tip of the iceberg, and whether longer cell-free DNA exists in the plasma. So we believe that one obstacle to the exploration of long cell-free DNA is the tool that we used for cell-free DNA analysis. So short-read sequences are commonly used in the analysis of cell-free DNA, but they can only produce reads of up to 600 bases. In contrast, for long-read sequences, they can produce substantially longer reads of more than 10 kilobases. So we set out to explore the presence of long cell-free DNA using long-read sequencing technologies. So first, we started by sequencing a maternal plasma DNA sample with both short-read and long-read sequencing. And we can see that with short-read sequencing, we can hardly see any DNA molecules that are longer than 500 base pair. But with long-read sequencing, we can see some long cell-free DNA fragments of few kilobases, and actually even up to 30 kilobases. And we found that as pregnancy progresses, the percentage of long cell-free DNA increases, and the median plasma DNA molecules greater than 500 base pair in the first trimester is 15%, and 20% for second trimester, and 30% for third trimester. So we know that the maternal plasma contains a mixture of DNA originating from the mother and the fetus. So to identify fetal and maternal-derived cell-free DNA molecules in the maternal plasma, we perform SNP genotyping of the maternal buffy coat and the fetal tissue. For SNP loci that were homozygous in the mother but heterozygous in the fetus, we can use it to identify the fetal-specific allele. And for SNP loci that were heterozygous in the mother but homozygous in the fetus, we can use it to identify the maternal-specific allele. And those plasma DNA molecules that carries the fetal-specific allele are the fetal-derived plasma DNA molecule. So these plots shows the percentage of fetal and maternal-derived plasma DNA molecule greater than 500 base pair in different trimesters of pregnancy. And this data confirm that long cell-free DNA from both fetal and maternal origin can be detected in the maternal plasma. So The analysis of long cell-free DNA has promising clinical potential because each long cell-free DNA molecule contains more genetic and epigenetic information than a short cell-free DNA molecule. For example, we have one SNP every 1,000 base pair in the human genome. So if we analyze a plasma DNA molecule of 200 base pair, the chance of it having a SNP is like 20%. But if we have a longer plasma DNA molecule, like 2,000 base pair, we can easily have at least one SNP in this long DNA molecule. So by using some computer simulations, we estimated that long cell-free DNA molecule in the size range of 1 to 3 kilobase actually has 5 to 6 times higher percentage of fragments with at least one informative SNP for differentiating fetal and maternal-derived plasma DNA molecule than the short cell-free DNA in the range of 50 to 600 base pair. As a result, using a less number, we can use a less number of long cell-free DNA molecule to attain the same coverage of the fetal genome than when we use the short cell-free DNA molecule. Apart from containing more SNPs, long cell-free DNA molecules also contain larger number of CpG sites than short cell-free DNA molecule. So this is one of the super long cell-free DNA molecules that we identified in our data. This 16 kilobase long cell-free DNA molecule covers 7 informative SNP sites. And with the genetic sequence, we knew that this molecule is coming from the fetus. And with long cell-free and with long read sequencing, we can actually deduce the methylation status of all the CpG sites on this long cell-free DNA molecule. And from our data, we know that maternally derived cell-free DNA molecule has an average methylation level of about 70%. But when we look at these long cell-free DNA molecule, its methylation level is 27.1%, which is much lower than the maternally derived plasma DNA molecule. So after looking at this data, we think that it might be possible to develop a method to differentiate fetal and maternal-derived cell-free DNA molecule based on their methylation patterns. So this formed the basis of the single molecule tissue of origin analysis that I will explain further in later slide. So after our finding of long cell-free DNA in the maternal plasma, we developed and we explored 2 potential clinical applications. The first potential application is on preeclampsia. And preeclampsia is a pregnancy-associated condition characterized by hypertension and proteinuria. And this condition poses substantial risk to both the mother and the fetus, and it affects around 5 to 8% of pregnancies. And we can classify preeclampsia into early onset and late onset based on the gestational age at diagnosis. So, in this study, we used long-read sequencing to analyze 5 early onset preeclampsia and 5 late onset preeclampsia. And for each preeclamptic group, we also analyzed additional 5 gestational age matched control. And from this graph, we can see that those preeclamptic patients actually have shorter fragment size distribution compared to the control. Specifically, there is a reduced percentage of fragments that is longer than 170 base pair. And by quantifying the percentage of DNA fragments greater than 170 base pair, we could differentiate pregnancy with and without preeclampsia. So here shows that when we used long-read sequencing, this classifier actually performed much better with an AUC of 1. But when we use short-read sequencing, the AUC is only 0.7. So the second potential applications is on the non-invasive prenatal testing of monogenic diseases. So when we perform long-read sequencing, we can actually obtain the methylation status of all the CpG sites on a single plasma DNA molecule. We hypothesize that by matching the methylation pattern on a plasma DNA molecule with the reference tissue methyls, we can determine the origin of individual plasma DNA molecule. For example, we know that the maternal cell-free DNA is predominantly from the maternal blood cells, whereas the fetal cell-free DNA is predominantly from the placenta. So by matching the methylation pattern of individual plasma DNA molecule with the reference tissue methylome of the buccal and the placenta, we can determine whether an individual plasma DNA molecule is more likely to be derived from the buccal or the placenta. Using this newly developed method, we achieve an AUC of 0.88 for differentiating placental and blood cell-derived cell-free DNA. And we also found that when you use longer cell-free DNA fragments, the performance of this classifier is much better, with an AUC of 0.91 compared to when we use short fragments, it's only 0.74. So next, we apply this single molecule tissue of origin analysis to the detection of maternally inherited mutations. We know that the detection of maternally inherited mutations has been more challenging than the detection of paternally inherited mutations because the mother, her own DNA is also released into the maternal plasma. So to demonstrate the feasibility of this approach, we perform this analysis in a family at risk for Fragile X syndrome. So Fragile X syndrome is a trinucleotide repeat expansion disorder, most commonly caused by the expansion of the CGG repeats at the 5' UTR of the FMR1 gene. So this mother of the family is actually a premutation carrier. She previously has a son who was diagnosed to have Fragile X syndrome, and she is now pregnant again with a male fetus, and we take a plasma sample from her at 12 weeks of gestation. We know that in the maternal genome, there are 2 X chromosomes, and in this mother, so one of her X chromosomes carries the premutation allele, and one carries the wild type allele. So we perform maternal haplotyping and link the heterozygous names nearby to this premutation and the wild type allele. For example, the C allele here is linked to the premutation allele, and the A allele is linked to the wild type allele. So we perform long-read sequencing of the maternal plasma DNA to obtain both the genetic information as well as the methylation pattern of the plasma DNA molecules. By applying the single molecule tissue of origin analysis, we found one molecule that is determined to be of placental origin and carries the A allele, suggesting that maternal haplotype 2 was passed on to the fetus, and the fetus would likely be unaffected. And this result was consistent with the diagnosis from chorionic villus sample. So this example has demonstrated the applicability of this long-read sequencing and single molecule tissue of origin analysis in the detection of maternally inherited mutations. So next, we further explore whether long cell-free DNA is also present in cancer patients and whether it can be used for cancer detection. So first, we demonstrated that there were comparable levels of long cell-free DNA in the plasma samples from healthy individuals, HBV carriers, and patients with hepatocellular carcinoma, HCC. So we also applied the single molecule tissue of origin analysis to cancer detection. And in this analysis, we determined whether individual plasma DNA molecules is more likely to be from the blood fecal or from the tumor tissues. We developed a scoring system which we named the HCC methylation score to determine the likelihood of a patient having HCC with a higher score indicating a higher likelihood. So we analyzed plasma samples from healthy individuals, HBV carriers, and HCC patients. And we can see that HCC patients have significantly higher HCC methylation score compared to healthy individuals and HBV carriers. We also found that when we use longer plasma DNA molecules, which have at least 7 CpG sites, the performance of this HCC methylation score could be improved compared with the use of shorter fragments with less number of CpG sites. So earlier this year, we also published a study to compare the performance of the two predominant long-read sequencing platforms, namely the SMART sequencing from PacBio and the nanopore sequencing for the analysis of long cell-free DNA in plasma. So for details, you may have a look at this paper. But here, I would like to summarize our finding on the tissue of origin analysis, because this analysis is central to some of the applications that I've mentioned before in this talk. We found that the performance of the two platforms are actually comparable in terms of performing the tissue of origin analysis. However, nanopore sequencing do provide 2 to 4 times higher number of fragments that are eligible for the tissue of origin analysis. And more recently, we also published a current opinion article summarizing what is known about long cell-free DNA and our future perspectives on this long cell-free DNA analysis. And you may have a look if you are interested. So in summary, long-read sequencing allows us to see a population of long cell-free DNA that was previously unseen when we used the short-read sequencing. And the analysis of long cell-free DNA has potential clinical utilities in prenatal and cancer testing. And the study that I have presented today marked a new era in liquid biopsy. With that, I would like to thank your kind attention.